Hello students, welcome to BioSmart Academy. Students, in today's lecture we are going to discuss about another part of immunology, that is immunoglobulin. This topic, immunoglobulin, is very much important from examination point of view. In the previous lecture I discussed mainly about the basic and some major concept of immunology. For example, I discuss about NK cell, I discuss about dendritic cell, I discuss about MSC molecule, both class 1 and class 2, both class 1 and class 2. These are the major portion that I discuss in the previous lectures. So those students who are still not see that all lectures, please go and watch that all lecture, because these lectures are very much important from examination point of view. So let's move to another lecture, that is immunoglobulin. Today we are start this lecture immunoglobulin, and this lecture will continue 2 to 3 days. Okay, so first of all we should know that what is immunoglobulin. What is immunoglobulin? Okay, before I start this lecture I want to recommend you that please share this channel to those students. Please share this channel to those students who are preparing for this examination. For example, CSIR, DBT, ICMR, GATE, etc. Share this channel to all those students, and please subscribe this channel. Please share each and every one, because this channel will provide you free lectures on every module. Okay, now let's start immunoglobulin. See, first of all we should know definition of immunoglobulin. What is immunoglobulin? Basically we know very well that what are antibodies. Antibodies are basically synthesized by B cells. Okay, exclusively synthesized by B cells. B cells basically synthesized millions of antibodies. Okay, millions of antibodies synthesized by B cells. Millions of antibodies are synthesized by B cells. Okay, and these antibodies are basically what? These antibodies are basically antigen binding glycoprotein. Okay, antigen binding. These antibodies are basically antigen binding glycoprotein. Okay, these are basically antigen binding glycoprotein. These are basically antigen binding glycoprotein. Okay, so basically antibodies are produced by B cells. B cells produce millions of antibodies, and these antibodies are basically antigen binding glycoprotein. Antigen binding glycoprotein. Okay, so and this millions of antibody that is formed, billions of antibodies that are formed. Each antibody have different amino acid sequence. Okay, each antibody have different amino acid sequence. Each antibody have different amino acid sequence and different antigen binding site and different antigen binding site. Okay, so each antibodies that are produced by the B cells. Okay, B cells produce billions of antibodies, and each antibodies that are produced by the B cells have different amino acid sequence and different antigen binding site, and these all are collectively known as what? Immunoglobulin. These are all are collectively known as immunoglobulin. So I think now you totally understand that what are immunoglobulins. I think now you have got a clear idea about the immunoglobulin. Okay, now see, now next move to the next topic. Okay, and basically these antibodies are basically Y shaped. Okay, these antibodies are basically what? Y shaped. Okay, now move to the next lecture. See, so here I just wrote it down. The things that I taught you, this all are written in this slide. Okay, antibodies, the antigen binding glycoprotein. Okay, synthesized by B cells, and each with different amino acid sequence and different antigen binding site. These are collectively known as immunoglobulin, and these are the most abundant protein in the blood. Clear? Next, see, basically antibodies are Y-shaped, as I told you earlier, Y-shaped, and this Y-shaped molecule have two identical antigen binding site. See, if I draw this in a bigger way, okay, so these two tip of the arm, these two tip are basically are basically antigen binding site. What? These are basically antigen binding site. These are basically what? Antigen binding. These are basically antigen binding. These are basically antigen binding site. These are basically antigen binding site. Okay, these are basically antigen binding site. Okay, so these two teeth or these two corners are basically antigen binding site. Okay, antigen binding site. And one more important thing that I want to discuss about these antibodies are, these are basically consist of 4 polypeptide chain. Okay, what? 4 polypeptide chain. This 1 polypeptide chain, this 2, this 3 and this 4. These are the 4 polypeptide chain that is consist. Okay, see if I draw it clearly. Okay, this basically consist of 4 polypeptide chain. Okay, 4 polypeptide chain. Okay, these 4 polypeptide chains, you can see it. Okay, and it basically consists of 2 identical light chains and 2 identical heavy chains. Okay, it consists of 2 identical light chains and 2 identical heavy chains. So what are the light chains and what are the heavy chains? These two, this one and this one are basically the light chains. Clear? And these two are the heavy chains. Okay, so consist of 2 light chains and 2 heavy chains. Basically 4 chains are there. Okay, and the connection between these 2 light chain and 2 heavy chains are the connected by sulfide bridge. These connections are basically sulfide. Basically sulfide bond is there, that is SS bond. Okay, that is SS bond. This connection consists of what? SS bond and other non-covalent bond. Okay, and I earlier told you that these are basically the what? The 2 tip of the corner is basically antigen binding site. These are basically the what? Antigen binding site. Okay, antigen binding site. These are basically what? Antigen binding site. Okay, so I think you have now clear idea about this structure. Okay, now, so basically in one word, if we say what are antibody, so basically antibody is a dimer of heavy and light chain. Okay, so antibody is a dimer. Antibody is a dimer of heavy and light chain. So antibody is a dimer of heavy and light chain. Okay, now, so this is all about a simple structure of antibody. Question may be asked from this topic, so this is very important. Please remember all these points that I taught you in this class and in this figure, because I will just explain with the help of this figure, so that when I read the points you have no doubt on that. Okay, now see, I already told that it is basically made up of 4 chains. Okay, 4 chains. See, these are the heavy chains and these 2 are the light chains. Okay, these 2 are the light chains. Clear? And these chains are connected by disulfide bond, that is SS bond. Okay, SS bond, disulfide bond. Okay, disulfide bond and other also non-covalent interaction are there. Okay, and I already told you that these 2 sites are the antigen binding site. Okay, that is this antigen binding site. Okay, antigen binding site. These 2 are the antigen binding sites. So clear till now? Okay, now it has 2 chains, that is these 2 are heavy chain and these 2 chains are light chains. Okay, and this light and heavy chain are connected through disulfide bond. So this is our immunoglobulin structure and these 2 are the antigen binding site. Now see, light chains are basically have 2 classes. Okay, light chains are of 2 classes. If we classify the light chain, see, I represent light chain with yellow. So basically it is of 2. One is kappa and another one is lambda. Okay, one is kappa and one is lambda. Okay, one is kappa, one is lambda. So these are 2 light chain, and any one individual of a species produce both the light chains, produce both the light chain. Suppose there is an individual of a species, suppose an individual of mammalian species produce both this type of light chains. Okay, but if we see any one immunoglobulin molecule, see if we see this immunoglobulin molecule, then we can find out that the light chain are either kappa or alpha lambda. Means in suppose this is an immunoglobulin molecule, in any one immunoglobulin molecule the light chain either kappa, if it kappa then it will be kappa only, if not kappa then it will be lambda, okay. So this is the main concept, that individual of one species produce both the light chain, but one immunoglobulin contain only one light chain, either kappa or lambda, but never both in a heterodimer condition they are present. Okay, either they contain both kappa or both lambda, but never in the form of kappa and lambda. Okay, in one single immunoglobulin molecule the light chains are present either in kappa or lambda, but not in kappa or lambda. Clear? Now next, basically as I told you that there are two light chains. Okay, but if we see the structure of heavy chains, basically it consists of mainly 5 types of heavy chain, 5 types. Okay, that is alpha, gamma, delta, epsilon and mu. These 5 types of heavy chains are present, and according to their name they are classified as a class of antibody, For example, alpha is IgA, okay, gamma is IgG, delta is IgD, epsilon is IgE and mu is IgM. So basically this heavy chain are classified into 5 different or major types of class of antibody, that is IgM, A, G, D, E, and these are came from 5 different types of heavy chain that is alpha, delta, gamma, epsilon and mu. So this is the basic concept of heavy chain, but we see earlier in case of light chain there is only 2 major classes of light chain, that is kappa and lambda, and an individual of consists both kappa and lambda, but in a particular immunoglobulin molecule either kappa or lambda is present, not kappa and lambda both. Okay, similarly in the case of heavy chain as well, an individual of a species make all heavy chain. Okay, suppose an individual of species make all heavy chains, an individual of species make all heavy chain. Okay, an individual of species make all heavy chains. Okay, but the major point is that if an antibody molecule is there, if an immunoglobulin molecule or antibody molecule is there, then either of the class of antibody will be present. Suppose an individual species make all heavy chains, but in any one antibody molecule both heavy chains are identical. Okay, suppose this is an antibody molecule, okay, and this is an total antibody molecule, okay, that is consist of both heavy and light chains, and if this antibody molecule is consist of IgG class, okay, suppose it consists of IgG class, then the structure will be kappa 2 and gamma 2. Okay, this will be the structure, kappa 2 or gamma 2 or lambda 2 or gamma 2. Okay, lambda 2 or gamma 2. So this will be the structure. See, an individual of a particular species contain all heavy chains, make all heavy chains. Okay, an individual of a particular species make all heavy chains. Similarly, an individual of particular species make all light chains, but in case of antibody molecule, only a particular class of antibody will be there, and particular light chain will be there, major class of light chain will be there. For example, if this is an antibody molecule, okay, if this is an antibody molecule make up of both heavy chain and light chain, and if this antibody molecule is belong to IgG class, okay, if this antibody belong to IgG class, okay, IgG class, then the structure will be what? That is kappa 2 and gamma 2. Why kappa 2 and gamma 2? Because IgG class, that why it is gamma 2 and kappa 2 light chain is present. Then kappa 2 will be there, not lambda will be present. If lambda is present, then okay, lambda is present with gamma 2, okay, then kappa will be absent. So in a particular antibody molecule, either one major class of light chain or either one major class of heavy chain will be present. Not all the major classes are present at once in a particular antibody molecule. So this is the basic concept that I discuss about the antibody molecule and the basic structure of the antibody molecule. Okay, so this lecture is all for today, and I wrote it down all the important points, okay, all the important points, and that I will discuss now only. Okay, first I think, and then your concept will be clear more. Okay, now let's read out these points fast. See, simplest antibodies is Y-shaped molecule, as I already told you, with two identical antigen binding site. As I already told you, that is, these are the two identical antigen binding site, as I already told you. Okay, now that's why they are called bivalent. Why bivalent? Because of the two identical binding site, they are called bivalent. Okay, now basically 4 polypeptide chains are there. Yes, 4 polypeptide chain, 1 2 3 4 polypeptide chains are there. 4 polypeptide chains are there, and it is a heterodimer, consists of light chain and heavy chain. Light chain's molecular weight is 25,000. Light chain molecular weight is 25,000 and heavy chain is 50,000. Okay, that is heavy chain and light chain, that is a heterodimer. Okay, now next, each light chain is bound to heavy chain by disulfide bond. Okay, by disulfide bond. That antibody is a dimer of heavy and light chain. Means these are the heavy chains and these are the light chains that are bound by a disulfide bridge. So the bond between these are SS bond. Okay, I already clear all these things with the help of diagram. If you understand, then it will be better for you. Okay, now all species have two major classes of light chain, that is kappa and lambda, as I already told you, and any one individual of its species produce both type of light chains. However, in any one immunoglobulin molecule, the light chains are always either both kappa and both lambda. Okay, both lambda. Okay, means an antibody molecule contain either kappa or lambda, but not in a heterodimer condition they will present. Okay, they contain either kappa or lambda. Okay, that is kappa 2 or lambda 2. Okay, next, now IgG, that is immunoglobulin of heavy chains, basically consists of 5 types that I already discussed with you. That is alpha, gamma, delta, epsilon, mu, and these 5 different types of heavy chains are called isotypes. Okay, isotypes means simply basically there is a difference in their amino acid sequence only. Okay, and because of their difference they are basically classified with these types. Okay, and because of these all types they are classified as a group of these, as I already discussed with you, and each class have either kappa or lambda, but not both of them. Okay, that I already discussed with you, that if this is a antibody molecule, okay, this is a light chain, okay, antibody molecule, then if this antibody molecule belong to IgG, then it consists either kappa 2 or gamma 2 or lambda 2 gamma 2. Okay, this will be the structure, not both of them are present in a particular antibody molecule. Okay, next, this is our last slide. Any individual of his species makes all heavy chains, as I already shared you, that any individual of his species makes all heavy chains, but in any one antibody molecule both heavy chains are identical, as I already discussed with you. There's an antibody molecule of IgG class could have the structure kappa 2 or gamma 2. So this that's all for today's lecture. Please subscribe my channel and share it with your friends. If you like my lecture, then please hit the like button. Thank you.